Hello students. So today we are looking at molecular basis of inheritance. So we will be attending questions from this chapter and we will have some questions and go through the explanations. Now this is a very important chapter in terms of whether you are preparing NEET or if you are going for any career where you are working with biology. This is a very important chapter because it deals directly with the genetics. The Severo-Ochoa enzyme is, this is a fact-based question and this is not, no, and this is our answer, polynucleotide phosphorylase. Okay. Now Severo-Ochoa, okay, and he won Nobel Prize for the discovery of these enzyme. Okay, and these enzymes, polynucleotide phosphorylase. Okay, phosphorylase, these are the enzymes which break down RNA into its individual components which are nucleotides. Okay, now naturally it breaks down RNA, but this is a reversible enzyme, so in under controlled condition it can also work to synthesize RNA, okay, without depending on DNA as a template. Alright. You are given a nucleotide sequence, okay, so we have a codon sequence. Which of the following is correct? Okay. Now it can be part of a DNA sequence. Acha, okay. So from the options we can see whether they want to know whether it's a DNA sequence or a RNA sequence. Alright. And if you look at the last option, which is an interesting one, the data is insufficient. So they also want to know whether we can deduce whether this is from a DNA or an RNA, right? So the major difference between DNA or RNA, which we can see from here, obviously there are more differences. There is difference in sugar and there is difference in nitrogenous base. Okay. Now if there is a difference in nitrogenous base, so what are the differences? So we know that in DNA and RNA, the major difference between in terms of nucleotide or nitrogenous base is DNA has a thymine and RNA has a uracil. Okay. Now we can see that T is present, so that means this option is out of the picture. We can clearly say that this belongs to DNA and it's not a RNA. Okay, so our best answer is A. Obviously this is not, and it can be part of either. No, it cannot be part of either. So let me know in the comments if you have got this question right. Also practice that you pause the video, get your answer and then see if you are matching with the correct answer. And more importantly, you should also know, okay, so there is a way to learn, okay, from the questions itself, that if you have seen that first answer is the correct, okay, that is well and good, but you should also know why other options are not correct, why they are incorrect. Okay, so that is where your knowledge will grow exponentially. Always practice that. Okay, let's move on to the next one. In agarose gel electrophoresis, the DNA fragments move with different velocities. Okay, so we have 4 statements and we have to see which one of them is correct. Now fragments move in proportion to the nucleotide length of the fragment. Okay, now DNA fragments, okay, so I will share with you in one of the posts how actual agarose gel looks like. Okay, when you do research, okay, you will come to know and learning and exploring so many different things, especially when you are working with DNA. This here is called well. Okay, I am telling you how agarose gel electrophoresis works. So here you put your DNA sample. Okay, and once you have put your DNA sample, it will run at, if there are different sizes and nucleotide lengths available in your DNA fraction, So they will run at different velocity, and after let's say 1 hour, you will see some of them have reached here, some of them have reached here, some of them have reached here. Let's say you have put another DNA sample here, okay, and it has reached only let's say up to here, and there is only one band, okay. So that means this DNA sample, what you have put in the well, okay, that has only one nucleotide fragment of this particular length, okay. Now how do we know how much length is there? So we add here what is called as marker or ladder, okay. Why ladder? Because when you run this alongside your DNA sample, it will run at different heights, and we know that this is already known that these are the fragments which are available in this. So let's say this is 1000 base pair, this is 2000 base pair, 10 kilo base pair. Kilo means 10,000. Okay. So we can see that this has run to the same height as this. So we can know that this is of length around 3500 base pair. Right? What about this sample? So let's call them A and let's call this B. So B sample has a DNA fragment which is of 3500 base pair. What about A? And let's call this 7500. Okay. So sample A has 3 fragments. Okay. And what are their molecular weight? So one is 7500 base pair. Another one, if you see, is around 3000 base pair. And this one is close to 1000 base pair. Okay. So there are 3 fragments. These are different nucleotide length. And obviously, if nucleotide length, let's say there are different nucleotide connected, okay, longer the length is there, molecular weight is also increasing, right? That is obvious. Something has more length, it will have more weight. Common sense. One more thing before we move on. As you can see, the smallest, okay, between these 3, this and this, let's change color for a while, this and this, okay, the smallest one was farthest from the starting point, okay. Now, all had started moving together, okay, but they have reached after 1 hour at different distance because the difference was in their velocity, right? Distance, actually you can call it displacement upon time, it doesn't matter here. So since time is constant, velocity is proportional to distance or displacement, whatever you want to say here. So that means more distance traveled is because of higher velocity. So that means this one has the highest velocity, this one is slightly slower, and this one is the least of all 3. Fragments move in proportion to the nucleotide length of the fragment. So that means nucleotide length is more than movement is less, right? So this is our incorrect answer. Fragments move inversely to the nucleotide length. Yes, this is correct. Fragments move in proportion to their molecular weight. No. Fragments move inversely proportional to their molecular weight. Yes. So the best correct statements are 2nd and 4th. So the best answer is C. Let me know in the comments if you have understood, or if you have any doubts. We'll take this question again. Let me know in the comments. Alright. Calculate the base ratio of a DNA sample if guanine was found to be 24%. Okay, now what is base ratio? Base ratio means A plus T divided by C plus G. From the Chargaff's rule, we know that suppose G is, not suppose, we have given this, so that means C is also this. Now if C is 24 and G is also 24, this is how much now? 48%. So that means the DNA has 48% of C plus G. That means A plus T is how much? Obviously it will be 100% minus 48%, and the answer is 52%. Right? So the base ratio will be this divided by this. Okay. So the answer is 52 divided by 48. Okay. Now let's see. You don't have to calculate. Always see, if you have a calculation which requires calculator, always see the options, and you may find a way to figure out which of these will fit without actually calculating it. Okay. Now obviously we know if numerator is bigger than denominator, the answer will be always greater than 1. Okay, now let's see which of the option is greater than 1. No, 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 yes. So our best answer is D. Always be smart when attending questions. Okay, you don't have to be, the perfect way to get the answer. Once you have solved it, just move on. You have, see, in your, for example, in NEET, you have to solve 90 questions from physics, chemistry, and 90 from biology. And what kind of biology I have written here? And 90 physics and chemistry. Okay, they will have some numericals also. Okay, so if total is your 3 hours, okay, you cannot give 1.5 hour to biology. That is not smart way, because then you will have less time for these things. Okay, so for 90 questions of biology, okay, allocate maybe around 70 minutes. Okay, remember this. In bacterial transcription, rho factor is, okay, so that's a fact based question. The best answer is termination. Okay, now remember this. This is sigma initiation. This is rho termination. Okay. This is the same letter you must have read in your physics. Okay, when you were reading electric current. Okay, so sigma initiation, rho termination. All right. So that's it for today. We'll connect on Wednesday with more questions on molecular basis of inheritance. All right. Stay safe. Have fun. Bye bye.